will have plateaued. So thank you all for joining us today for the ACS Physical Division webinar, generously sponsored by PHYS. My name is Rodrigo Noriega. I'm at the University of Utah, and it is my pleasure to introduce Dr. Andrei Tokmakoff, the Henry G. Dale Distinguished Survey Professor at the University of Chicago Chemistry Department. Andrei and his lab have developed techniques and analyses that have really become the gold standard in the field of condensed matter dynamics. From water to proteins, as we'll hear about today, his studies with structure-sensitive tools have shown the inner workings of protein conformation as well as dynamic hydrogen bond networks. And his amazing contributions to science have already been recognized in many venues. He was recently elected to the National Academy of Sciences. Congratulations. He has also received numerous awards and distinctions, including the Errol Plyler Prize for Molecular Spectroscopy and Dynamics from the American Physical Society, the Ahmed Zewail Award in Ultrafast Science and Technology from the American Chemical Society, and the Ellis Lippincott Award from the Optical Society of America. And today we're going to hear about his work on site-specific binding dynamics by proteins. During the seminar, as a reminder, we ask you that you please type your questions into the chat in your Zoom window, and then we will collect them for a moderated Q&A session at the end. So please join me today in welcoming Professor Andrei Tokmakoff. And Andrei, the floor is yours. Thank you for being here.
Thank you. Thank you, Rodrigo. And actually, that goes for Lou and Vanessa and Marco also. Thanks for giving me the opportunity to speak with you. It's a very nice introduction, Rodrigo. And after that, I'm afraid everything that comes afterward is going to disappoint. I was just telling our organizers that I'm very nervous because this is a topic I haven't talked about, before, and it's not entirely complete, but it's more just, it's a problem that I'm just fascinated by and wish to get more insight and encourage other people to study it. So the topic is the dynamics of molecular recognition and binding in protein-protein interactions. And this process, it's remarkable for a number of reasons. You have two protein binding partners, and they encounter one another diffusively, and they interact first non-specifically, but then in some manner locate and find site-specific contacts. And it's not unusual for these proteins to have large amplitude conformational changes. And in fact, it's very common that one or both partners are disordered and may sort of fold and bind in a concerted fashion. And the speed of these processes, how fast they are and efficient they are is, quite amazing. And these problems remain very difficult from a dynamics point of view to study experimentally and computationally, actually. So my group has been working for some time trying to develop experimental techniques to look at these dynamical processes in protein-protein interactions, also in DNA hybridization. And it's a complicated problem. And I'd say some of the people in my group probably also, like me, find it very hard to publish things in easily digestible pieces because there's so many parts to the problem. And so, what I'm telling you is really more of a progress report than a nice, clean message. But I guess the other message is that this is an example where developing new method really goes hand in hand with understanding the problem. So what I will be telling you about is insulin dimer formation. That's what this image is of. This is a encounter complex, nearly formed insulin dimer. And we feel that this is a nice model system that captures all of the complexity of these problems, and I'll tell you about that. But before that, I just want to introduce the problem a little bit further, and this is basically to tell you why I think it's fascinating and why other physical chemists should work on it. So what is the problem? Well, basically, the question is, how do proteins, binding partners, recognize and bind a target site specifically, at incredibly high rates, often in the presence of many other possible partners. Right? These terms I've put here, diffusion, encounter, recognition, binding, that's not sort of anything rigorous to those terms. I'm just using that to get you to think about the multi-scale nature of this problem in time and in space, and emphasize the shift, from non-specific long-range interactions to short-range specific non-covalent, contexts. And the other thing I'd like you to recognize in this process is, beyond biomolecular, macromolecule association problems, recognize that this is more generally a molecular self-assembly problem. And so physical chemists really are required, in order to gain true insight into these problems. Okay, so I thought I'd start by the thing that got me interested in this problem initially, and that is a set of experiments that are about as old as I am, and this had to do with looking at measuring the association rates for lac repressor binding to DNA. And what happened in this particular case is Briggs and Cohen, they measured association rates, for this lac repressor trying to find its operator, and they found that it couldn't be explained by three-dimensional diffusion. The rates that they were measuring were faster than what you would expect, and that is partially because what they were relying on was for their reasoning was basically a nearly century-old equation here about diffusion limited reaction rates, treating diffusion processes from solutions to steady state solutions to the diffusion problem, the flux at the boundary of a sphere, right? And that's just not modern chemistry, and so we don't really know what's going on. But part of the reason this was intriguing is it basically was sort of the stepping point to the area of facilitated diffusion. It got people to think that really it's actually a combination of one-dimensional sliding and three-dimensional hops that actually allow you to make sense of this association rate and the dimensionality being such an important problem. And this is where actually visualizing and getting clear observations of what's going on is important. This stuff is very different than what you would see now in simulations. And actually, it's only very recently that we've been able to do unbiased, real-time molecular dynamics simulations of a binding process. And this, it's a hard problem if you want to do all atom simulations with explicit water. Really, I'm really drawn to David Shaw's work in this area. This is where the two movies are from. He really does visualize things nicely, and they give you a sense of how much different this is than a random walk problem. Diffusion, for instance, on the left looks like two-dimensional diffusion on the surface of a drug on the surface of a enzyme. So these kind of what's happening structurally is what we'd like to get at experimentally. Let me just review the timescales and distance scales involved. Typically in 100 micromolar solutions, molecules will encounter one another every few microseconds, and first are interacting with long-range electrostatics. Once they come into proximity, this is where one thinks of the system as probably existing in an encounter complex that may involve both fleeting contacts or long-lived metastable states. You may explore many different configurations before finding a favorable binding site, and so there's problems like including what I call registry, so the mutual orientation, translational degrees of freedom of the two partners. You also have to worry about the conformational changes that are going to happen during this phase, and the water and ions involved, de-wetting of protein interfaces. And there's these conformational changes can involve very large amplitude motion, including folding. And we have to worry about depletion forces and all. So being able to visualize these things are what we have to, and then finally, there's the short range interactions. These sort of include both electrostatics as well as hydrogen bonding, hydrophobic packing, really trying to find a specific type of contact that will really stabilize the complex. So the question is, how do we get at some of these things experimentally? Well, it's hard. We want to be able to visualize some parts of these processes with structural insight and really be able to access the timescales of these processes, and hope from what you saw that you see that it's the nanosecond to microsecond time window that's particularly important. These are the timescales over which proteins move relative to one another, and reconfigure themselves, how water moves, and how molecules encounter each other. And so for that reason, your typical mixing experiments are just not going to work, because on a macroscopic level, the diffusion is so much slower, than the time scale for the interactions in this encounter complex. Magnetic resonance is really a powerful tool, but that's I tend to favor techniques that really look directly, and so the magnetic resonance has done amazing things. Although I categorize those techniques as indirect. One thing that is really coming along now is single molecule methods. It's not really quite there yet. These binding dynamic processes are in active development. The problem is really have to be fast, and that's something that single molecule methods are, have not quite gotten there. But I am particularly impressed, for instance, by this work from Hui-Sung Chung's group, who's really doing detailed photon emission statistics in 3-color FRET experiments to sort of deduce what's happening in binding and dissociation events. So there's a lot of promise there. But of course, single molecule fluorescence has the structural sensitivity issue to deal with. So our approach to this is really to ask the question about, do we have to actually watch the association process in order to learn what we want to learn? Can we do dissociation experiments using temperature jumps? So temperature is a great control variable for protein-protein interactions, because these are, delicate balances of, well, from a thermodynamic point of view, large and enthalpic and entropic terms with a delicate balance between these two dictating whether things bind or not. And so temperature can swing equilibria dramatically. Here's an example of a melting curve that similar to what you'd measure experimentally, in which you start with a complex and ramp the temperature, and the fraction of molecules in dimer form, can change drastically over only, 10 or 20 degrees. And this sort of gives you the, the sense for, the possibility of using temperature jumps. These kind of melting curves can really tell you all the, a lot of the thermodynamic information you want. And so the question is, what about the kinetics? So here we're typically doing 15-degree temperature jumps, and you can see, how drastically this shifts the equilibrium. So the types of experiments we're trying to do, they're not your typical ones, right? So here's what I call typical. You imagine that you're sensitive to some type of a dissociation process, and at the initial temperature, there's an equilibrium established here. You're primarily in the dimer form, and there are exchange kinetics between the dimer and the dissociated monomeric state. And a temperature jump shifts this equilibrium a little bit. And then it adapts again by the activated barrier crossing, between these processes. And you can, describe the kinetics of these processes pretty well. What we want to do really is to try and create conditions in which the temperature is far from perturbative. It's such a huge reshaping of the energy landscape that it creates barrierless conditions in which you really can just diffuse apart with minimal or no significant activation barriers. So, to illustrate this, we imagine, can we, shift this energy landscape so dramatically, that one has at least some significant fraction of the population diffusing apart. So that's the concept. The question is, does it work? And then does it tell you about the association process? We think that this will be able to create some of these encounter complex species and allow us to deduce things. Now we have to watch this process structurally. And so for that purpose, we're turning to infrared spectroscopy, both in 1 and 2 dimensions. And we're looking at the amide 1 vibrations of these proteins. So for those you don't know, the amide 1 vibration is the carbonyl stretch of the polypeptide backbone. People have known for quite some time that it is sensitive to secondary structure, although it's a pretty featureless infrared spectrum. But the basic concept behind all of this is that the arrangement of these carbonyls within secondary structures actually leads to strong couplings between each of these amide residues, and that leads to delocalized vibrations that extend over large parts of the protein. So when you're looking at infrared spectra, amide 1 spectra of proteins, these tend to be really quite delocalized, quite mixed, and sensitive in a global sense to what the protein is doing. Two-dimensional spectroscopy, it gives you a little bit extra. Still relatively featureless, but I tend to think of these as having a somewhat enhanced fingerprint of sort of a global fold of a protein. But it does have secondary structural information. And here, these are experimental examples of something. It's a bit idealized in terms of beta sheet and alpha helix content, but it gives you a sense for the kind of signatures we can look for. I'll just note to you while I'm looking at it, for those of you that are not familiar, the resonances in these 2D spectra are always one of these little positive negative doublets. Okay, now these delocalized vibrations aren't necessarily what we want for this problem. In fact, a large part of it is really trying to understand different types of contacts within the binding partners and between the binding partners. And for that, we really want to have local probes also. And this is accomplished by doing site-specific isotope labeling of different amide carbonyls. So, the basic idea is these carbonyls, their frequencies respond to forces. Forces can either stiffen or soften the vibrational potential, and this changes the vibrational frequency. These vibrational frequency shifts, they are useful for probing, hydrogen bonding contacts and electrostatics. But what makes them really useful is the fact that there's a real quantitative way of describing what's going on. And this is by thinking about the vibrational frequency as being an electric field, thinking about the CO stretch as being an electric field sensor. So, this is sometimes gets called the vibrational Stark effect, but basically the idea is really carbonyls are quite well behaved and they have fairly well described frequency shifts, linear in the sensed local electric field. And this forms the basis for computational modeling. And in fact, they are quantitative enough that we really can draw structures from MD simulations and calculate, infrared spectra of these labeled sites. So this really, draws from a work of a community of people that have been working on what's called spectroscopic maps that can be used to translate the instantaneous configurations you get from an MD simulation into vibrational frequencies, or actually amide 1 Hamiltonian that can be used to model the vibrations of a protein. I can't go into the full model here, but this is the basis for our interpretation. And these examples here are meant to give you a sense of the accuracy. So this plot on the left here, it shows you a comparison of on the y-axis, the experimentally observed frequency for amide carbonyls in small peptides, a variety. And the x-axis is the electric field on the carbonyl oxygen along the CO bond, calculated from CHARM MD simulation. And next to it, these are examples of simulated spectra for site-specifically labeled sites within a protein whose structure is very well known. So it's a very effective tool. Okay, let me turn now to our model system, insulin. It's I'm sure you have heard about this a lot. So as far as regulating glucose in your bloodstream, that's usually what we think about it for this. It's a kind of an unusual hormone, or in the sense that it actually has two different peptide chains, the A chain and the B chain, and these are actually held together with disulfide bonds. So it's not it's made actually synthesis of insulin incredibly complex, as all of these post-translational modifications. So thing is, insulin is actually stored primarily as a dimer or as a hexamer, that is a trimer of dimers, and these dissociate, in your bloodstream into the monomeric form. And this equilibrium between dimer and monomer is actually one of the things that that most insulin drugs are trying to control. That is, they're typically insulin mutants that are trying to change the equilibrium to favor the monomeric form in such a way as to make it more available. Now, this monomer structure is this. What I'm showing here is the crystal structure, but the truth of the matter is the monomer structure is not actually known, at least under normal conditions. So all of the solution structures, I mean, they're either crystal structures, which are in the dimer or hexamer form, or they're solution NMR structures in which you've had to do things to destabilize the dimer. High concentration leads to dimer or to aggregation. And so you add things like acetonitrile, or you make mutations to the chain. But there's a lot of unknowns, and particularly with regard to this B chain. The B chain C terminus here is of interest to people because it's thought, it's quite certain. It hinges open, and it is what binds with insulin receptor. And so there's a lot of interest in really what's happening conformationally with this B chain. So now it makes a dimer. In the dimeric form, as you see from a crystal structure, what you have is two monomers come together, and these B chains form an intermolecular beta sheet between them. So this is the first element that stabilizes them. There's 4 hydrogen bonds between residues of in this anti-parallel sheet configuration. But these are also aromatic side chains, and it's sort of the hydrophobic packing between this, what's known as aromatic triplet, that also acts, to give you extra stabilization of packing these side chains. And so it's this combination of these hydrogen bonds and weak packing, weak intermolecular forces that are holding this dimer together. All right, so what do we want to learn? We want to learn something about insulin as it influences the phenomena we talked about. But with every dynamics problem comes structure problems also. So we want to learn about the structural ensemble, of insulin monomer and dimer. We want to assess, whether a temperature jump can control this in a time-dependent fashion, and whether we can actually see the types of conformational and contact changes we expect. Some years ago, we did some test experiments. So I just want to show you some proof of principle experiments we did early on, just to convince ourselves that this wasn't an entirely crazy idea. And this was, in this particular case, we decided to make our life easier and work with bovine insulin and add ethanol. So this little bit of co-solvent in order to destabilize the dimer. As it turns out, any change at all to the buffer solvent conditions has dramatic effects on the behavior, and you may get a sense for that as we go on. So here are typical types of equilibrium characterizations. These are temperature-dependent IR spectra. The changes seem very subtle. In this different spectrum, you may sort of make out this little shoulder on this featureless spectrum, and it might be a little bit more obvious to you as this ridge in a 2D spectrum, and that is a sign of the anti-parallel beta sheet in this complex, and that goes away as you raise the temperature. This in 2 dimensions, it's only slightly more obvious, and you can essentially see these cross peaks from beta sheet modes that have gone away on melting it away. We can look at these kinds of experiments and deduce the dimer fraction and the equilibrium constant, enthalpies and such, and so there's many routes to doing the equilibrium characterization. Going to temperature jumps, here's how the experiment works. We use a laser temperature jump to ramp. Basically, weak absorption by water allows us to ramp the temperature. Maybe the way to think about it is we put a lot of kinetic energy into the water, and that thermalizes pretty fast. And we get about 10 degree, well, between 5 and 20 degrees possible temperature jumps. And they rise, temperature rises in about 10 nanoseconds or so. And that that non-equilibrium state is pretty persistent, pretty constant up until about 100 microseconds, at which point, there really is kind of a gradual cooling of the sample. But during this process, we come through and use IR spectroscopy synchronized, to monitor structural changes. And we can do this as IR absorption, regular. We can do this as any sort of, I'm sorry, don't know what happened there. Any time resolved, we can use 2D IR to probe what's happening and such. So just so you know what we look at, an equilibrium resonance in 2D always looks like this plus minus doublet. And that means once you start temperature jumping, what you're actually measuring is the transient difference spectrum of what changed relative to equilibrium. And so these are what loss and gain features would look like. And so here's an example of some of these transient 2D spectra. The equilibrium spectrum is right. What changed after a given waiting time, 10 nanoseconds, 10 microseconds, so on, shown over here. And it's maybe a little easier to see what's happening if I take away the contours. And so these experiments showed us that, indeed, as you move into the 100 microsecond region, you're starting to see a clear loss of these beta sheet features that were sort of beneath this AMID1 spectrum. So that was really nice. That gave us a lot of confidence that we're seeing dimer dissociation. And we can look at these kinetics. Our strategy for doing this, because those spectra are so long to acquire, is we actually do a slight variation on that, which is a transient absorption probe. And so that's equivalent to projecting the spectrum onto the detection axis. And so here are typical changing spectra as a function of this T-jump delay. And we can use these to extract dissociation kinetics. And we repeat these for many different initial temperatures. And we do have control over initial and final temperatures. Okay, so now what we do is we go, study these as a function of concentration and final temperature, and do sort of a global, analysis of all of these data sets using a reconstruction method based on constraints applied to a singular value decomposition. And the net result of all of this is shown you here. What we find is after removing contributions due just to the heating of the solvent, and there are two dominant changes to the protein spectrum that we observe. And I'm showing you the kinetics and also the spectrum associated with those kinetics. When you look at these, what you deduce is that there's this relatively slow process. And by that, I mean it varies between 250 and 500 microseconds. It follows nice exponential kinetics, and it's really correlated directly with beta sheet loss. And therefore, we associate it with dissociation of the dimer. There's also something that precedes that, or it overlaps with it also. It spans 5 to 150 microseconds or so. And that, from the spectrum, is a different part of this AMID1 band, and we attributed it to alpha helix conformational changes. That's essentially what the spectrum was suggesting to us. And so that, just these assignments were also, we came by them a little bit also just by doing experiments in even more ethanol, which really dissociates the dimer entirely. And we lost the first component and kept some of the second. So it may be some sort of an alpha helix unfolding. These experiments gave us the confidence that we can do this properly and invest the time and effort and money to do this the real way. And so the basic idea is we want to move beyond the standard thermodynamic kinetic investigation that treats everything like a two-state, all or nothing process, that they're either fully separated monomers or perfectly intact dimers. But we know once you start looking at a molecular level, it's not going to be that simple. But once you start really looking at specific contacts and conformational changes, it's probably not going to be far more complicated. So our first step for this was to select some isotope labels to help us with this. And these were actually designed using some of the computational spectroscopy methods I told you, but we basically looked for a number of different sites that could be used to probe specific non-covalent bond contacts or hydrogen bonds in different regions of the protein, both at the interface and away from it. And we inserted site-specific C13018 labels into the backbone. And this was done by a heroic postdoc, Paula Dialon. If any of you know about the history of chemical synthesis of insulin, you'll realize this was amazing. It's a full chemical synthesis of this in order to get the site-specific positioning, and really heroic by itself. But we ended up with a number, of labels, and we do these either as a single isotope label to sort of sense the electric field near that carbonyl, or sometimes as dual labels where they're oriented along a shared hydrogen bond. And that really gives you an additional coupling effect to look at contacts and folding. And the way we look at this typically is through an isotope difference spectrum. So here, this is the example of what happens. On top here are the regular infrared spectra of the unlabeled insulin, UL, and the isotope label, in this case, a dual B24, B25 label. It was right at this dimer interface in the beta sheet. This is one that we're going to see a lot. We take the difference of those two, and so it's this isotope difference spectrum that we're really using. You can see that here, several tens of wave numbers redshifted from the band. We've pulled out the vibration associated with those labels. We also have these two loss features. I just want to point out that's a clean little doublet of sharp peaks that are lost that really look just like a beta sheet. That's what we were seeing earlier. So now we can go with all of these labels in different regions that are meant to sense different parts of the protein, and we can measure spectra here, first of all, at low temperature, where we really are entirely in the dimer form, and in this label region, you can see a large variation in the peak positions for this label, and there's some odd line shapes in there also. And the variation in these frequencies is really very broad, much broader than you'd expect. You can then ramp up the temperature, and this essentially will give you the spectrum of the dimer once you're at a high enough temperature. That forms a basis, at least for understanding, well, at least a high temperature monomer structure. Okay. We can look at the melting behavior of each of these peaks in such a way as to, get a sense for how different parts of the protein are behaving. And these are really, just some mildly corrected traces right along, these peak frequencies. And they're actually, they're really pretty hard to interpret just by themselves. But I'll point out, a couple of things. First of all, the real dissociation of the dimer, which we think is measured by this main amine-1 band, tracks this dashed line, and it when you compare all of these other labels, you see they all behave a little bit differently. They vary in their slopes and where they, their maximum inflection points and their apparent melting temperatures. And so there is some, even though they coincide a little bit, there is some variation. So let me just sort of give you, talk you through a couple of examples of the kinds of things that you can learn. So first of all, let's focus on 3 labels. These are selected, in this way. So the first one is what I just showed you. That's this B24, B25 residue, and that leads to this double label on one chain leads to 4 oscillators that are all coupled right in this beta sheet that forms the interface. It's a very good sensor of this dimer interface. Quite different is this A13, A16 label. It is in the helix here. We call it the AC helix just for being at the C terminus of the A chain. It's not involved in the interface or apparently at all. And this double label really just looks at how folded is that helix. We also look at the turn. There's a double label B20, B23. This is in certain configurations of this turn, there's a hydrogen bond between those 2 residues. And we'd like to know what's happening there because that's where the hinging motion, of this chain is. When you look at the melting curves for those, and this is normalized and baseline corrected, and but it's really just sort of a measure of spectral amplitudes. You can see they vary quite a bit amongst them, right? So the dimer fraction, that's what you see in black, it overlaps fairly well with this B24, B25 label, which is supposed to see dissociation, but they're not the same, right? They do behave differently. This B23, B23 is very gradually evolving over a large temperature range. And that's A13, A16, it only changes at high enough temperatures that you're almost all monomer. So this is probably thermal unfolding of that helix, even in perhaps only in the monomer form. So, this is interesting. It sort of raises this question about how correlated is the dissociation event from the changes in stability at different parts of the spectrum. And if you're sort of thinking about this kind of language is reminiscent of what you think of as cooperative binding. So usually that's done in different contexts, but think about using these kind of models. In this case, this is a weak binding partner. You in fact, we know the dimer is bound by a number of different contacts, many of them aromatic clustering and a handful of hydrogen bonds. If indeed there is sort of a modest energy benefit to making one contact and a higher propensity to make a second contact adjacent to it, then you might expect to get slightly different melting curves for the contact as opposed to the global behavior. And this is a simple Pauling model that got used in the ancient days of early describing, allosteric effects in hemoglobin. But you basically, can see how you think an ensemble will evolve based on these sorts of cooperative phenomena. So the blue line here, this is just a few kcal stabilization for each contact plus these cooperative benefits for adjacent contacts. The blue line is those fraction of molecules that have all of their contacts, all 10 of them in place. Right? And you can see this has a very steep melting curve. But the fully monomeric form where there's no contacts, this is really a bit more complicated. And it's because, within this intermediate range near the melting temperature, there are, of course, when you've got weak interactions, it's always a possibility that you're going to be missing one or two contacts, right? Or maybe even at elevated temperatures, there are things that have, be it transient or not, one contact, And so I think this kind of a picture probably, when we look at it a bit more closely, will have something to do with what we see here. All right, so now let's do this, but in a temperature jump mode. So here we're going to just jump right across this main dissociation temperature. This is just like I described it to you before. We're looking at projections of this 2D spectrum onto this detection axis. These are the spectral changes you see. Here you can, down in this corner of the 2D spectrum, you can see the changes to the labeled region in this particular sample. And we see that through the changes in this peak over here. And so as a first panel, here are the kinetics associated with the 3 labels plus the unlabeled sample. In that region, what do we see? Well, the unlabeled sample, it has a response, but it perfectly tracks the temperature of the sample. So it relaxes, but it relaxes as thermal diffusion carries the heat out of the sample. The alpha helix, it has a very little response. I really had to scale it up by a factor of 3 just so you could see it's doing something. So it doesn't seem to be really responding, much at all. This B23 turn, it's only mildly better. But we see a nice, clean, well-behaved exponential behavior for this interface, the B24, B25 label right at the dimer interface on a 290 microsecond time scale. So that is really a clean metric for the dimer dissociation in the sample. We can now go look at other points in the main band. So that's looking at other regions here. And what we see, looking at these different samples, so the unlabeled, besides the unlabeled, you should be thinking that the main band of labeled samples really are a different spectrum than they were before the label, because you have essentially removed and deleted certain oscillators out of it. And so what you can see is all of these responses are much faster than dissociation. They are very stretched and non-exponential, and they span many decades in time preceding the dissociation event. We can put that on a more quantitative footing. And what we find is that of all the kinetics we see here, the slowest is the dimer dissociation, and the most exponential is the dimer dissociation. That's these green spots over here. The rest of these relaxation processes are shorter, in some cases much shorter in terms of their kinetics, and considerably more stretched fit to that functional form. Okay, so what does all of this mean? Well, in essence, what we think is that there is considerable dimer reorganization prior to dissociation, that there, amongst all of these weak contacts, there is considerable room for organizational changes, and that these conformational changes in some way precede the dissociation. What could that be? All right. That's sort of one of our first questions, is how do we think about what's happening, in those kinetics? Before we're going to interpret that in terms of modeling and simulations, let me give you sort of the second problem that came up where we needed some simulation insight. And this was looking also again at this label at the dimer interface I told you about before, a odd result that was unexpected that suggested that maybe there's different structures to this dimer. So when you look at this labeled part of the difference spectrum, there was quite a bit of time where we had lots of problems with reproducibility, and gradually figured out every small detail about how you prepare the sample is going to give you different results. And so this is one example, is that the salt content of the and buffer salts and things like that, co-solvents, anything will change things around. But in this case, a high salt, low pH condition led to 2 peaks where there was only one when you were really at low ionic strength. And in fact, then when you melt these dimers, you find that these 2 peaks of the dimer form actually melt differently. That this one here in the center at 1592, it melts away first before the other one. You can see it a little bit in 2D here, that at this lowest temperature, the central peak is the strongest, whereas the relative ratio of the two is changing even well below the melting temperature. Okay, so there seems to be some kind of dimer equilibrium. Could it be? Yeah, and with some other experiments I can't tell you about, Curie actually determined that this was really an ionic strength effect that follows what you'd expect, for electrostatic screening. The ionic strength dependence really follows the Debye-Huckel limiting law for the ratio of these 2 features. And that seems reasonable, because I didn't mention that, this is at a low pH. I think this was taken at pH 3, and to sort of help stabilize or help solubilize the system. And at that pH, the net charge on an insulin monomer is +5. And so it stands to reason that really you're going to stabilize the dimer, and perhaps different forms of the dimer, with increased ionic strength. Okay, so I've posed 2 kinds of experimental insights that didn't quite understand, and I just wanted to turn briefly to simulation to tell you about what's how we're trying to answer those questions. Some of these come from working with Vijay Pandey on the dimer structural ensemble, and then with Aaron Dinner on the monomer structure and dissociation mechanism. And for this dimer structure, Vijay's really extensive unbiased MD simulations of the dimer were used to build a Markov state model of this dimer. And this network plot essentially shows you the nature of this model. The radii of these nodes is related to their population and their proximity and their line, and the line strength connecting them tells you about the kinetic rates, of exchange. And when you color code this by RMSD, you really clearly see that there's 2 kinds of clusters. And we've called these blue ones native. They are closest in RMSD to the crystal structure. This other one is twisted because, at a glance, it just seems like you twisted the interface by about 50 degrees clockwise. And in fact, these 2 sides of these states, that's how they differ. One of the things you've done is in twisting this is essentially to rupture, the beta sheet, but you've not changed it entirely. You've only shifted the registry of the beta strands. You've lost a couple of hydrogen bonds, and the orientation of the 2 helix packed at the interface is rotated, and the sidechain packing is still efficient, but very different than before. So That's these two states. And the rest of these states, they, fall more into the category of having varying degrees of disorder, where different parts of the helices, the AN helix, the one at the N terminus of the A chain, is melted on one or both sides of the monomer of the dimer. We look, these Markov state models, they encode the kinetics and give you some insight into the dynamics of the system. When we really look at the slowest time scales obtained by this TICA, time lagged independent component analysis, it sort of associated the slowest 14 microsecond time scale for the exchange of population between this native and twisted state. There's some other poorly behaved states in between these, and thinking of those as intermediates, we sort of coarse grain this down further. And the kinetic network here really demonstrates pretty clearly that the dominant path to exchange between these native and twisted forms is to go through two states where this beta sheet is solubilized. So you actually have to rupture this beta sheet, get some water between it in order to enable this rotation. Generally speaking, there's a lot of disorder and fluctuations in each of these wells. We can then go to all of these Markov states. We can calculate IR isotope difference spectra for all of them, and we can begin to lump these both in spectrally similar and kinetically clustered regions. And all of this forms a basis set, which we could use for ensemble of refinement. We haven't done it yet, but that's very much our intent. And when we go look and do calculations for the average of the native and twisted state, we find that the isotope shift that our calculations predict are reasonably well in line with what's expected, for the two features that we saw in the high ionic strength. So that's our interpretation of what's happening at the dimer interface. All right, similarly, we did something similar for insulin monomer, this time with the help of Aaron Dinner, and found the predictions that there's a lot of disorder expected for the monomer, both in detaching of the different termini of these chains and the melting of this AN helix. And there's some IR evidence we found for that. This B6 label, it can exist in a solvent exposed or a buried form. And what we found is that the melting of this isotope, oops, sorry, my screen just went blank.
Okay, we've had some connectivity issues. We'll wait a minute or two, and then we can see where we stand. Thank you all for your patience.
Even after all this experience and time with Zoom, there's always still, things that can happen.
Folks, I'm sorry to say the weirdest thing ever happened. Hope you can hear me okay. My desktop just randomly shut itself down. So I'm joining you on my laptop real fast. But I think that shutdown was prescient. I was prescient. I was getting a little bit late in my presentation anyway, and not so much to do. So I would say I'm going to call it quits, but there is actually a very important slide that I forgot to show you. So I will put that one up. Yeah, the people that have were brave enough to join this project and have contributed over the years to parts of it as it moves along. Sorry, I won't be able to tell you a little bit more about some of the modeling of the actual mechanism of association and dissociation that Aaron Dinner's group has helped us with. But at least some of that's published. So most important for me to acknowledge Qirui Feng. He is the one common factor in everything I told you. He's really had his hands both in experiment and the simulation and modeling, and has been unstoppable in trying to deal with challenges. Evan Jones did the original proof of principle experiments. And Paul Sandstead helped with the more recent labeled T-jump experiments. Luis has been in charge of some of the modeling also. Aaron and Adam, they were the heroes that did most of the work on the modeling for us, but also with Bodhi. Steve Kent is the world's greatest synthetic protein chemist, and I don't think I'm exaggerating. If you want to hear some stories about how he synthesized a full D-stereochemistry protein, I can tell you. And Bala was the one that made all the samples we talked about. I want to thank all of you besides that for your attention, and happy to answer questions.
All right. Thank you again, Andrei. We'll jump into the questions right away. We have one from Georg Zimmerman with a question about the insulin melting experiments. And the question was, so what is the midpoint temperature for the dimer monomer transition and for the thermal unfolding? Both should shift the signals in a different way, with melting perhaps being the bigger effect, and how is this deconvoluted in the experiments?
I'm sorry, Rodrigo.
Sorry.
I only unmuted things halfway into your question.
All right, no worries, I'll start again. So this question comes from Georg Zimmerman with a question about the insulin melting experiments. What is the midpoint temperature for the dimer monomer transition and for thermal unfolding? Both should shift the signals in a different way, with melting perhaps being the bigger effect. And how is this deconvoluted in the experiments?
It's, from an experimental point of view, it's a very important question. And you probably won't be very satisfied with the answer. The challenges you're talking about, they are right. The ambiguities that exist are always there. And so melting experiments have always got to kind of keep that in mind, that you can't read too much into them, because so there's so much uncertainty in trying to determine what effects are simple thermal shifts and what are real structural changes. One of the advantages, that we have is, we have certain real structure specific markers that, allow us to, distinguish certain particular types of structural changes. But any one particular experiment is totally vague. So this is part of the reason why I don't spend a lot of time talking about it, because to do a real analysis of a melting experiment really requires a self-consistent analysis of many experiments, so that any one doesn't matter. In our case, we would really like to be able to describe all of our isotope labeled melting, all on the same, all on the same basis, and and rooted in sort of correlations with structure. But I also trust things that involve trends, for instance, with ionic strength or co-solvent or insulin concentration. These are all other strategies also. And kinetics ultimately is more, insightful, because then you really have both the structural spectral marker as well as, a clean time scale associated with it. Hopefully it makes sense. But, your concerns are totally justified.
I think there's a follow-up question to that one as well that just appeared in the chat. Do we want to address that or go to the other question?
I guess we can bring that comment in. So the comment as a follow-up is, perhaps you could start the temperature jump at lower temperatures to not unfold the protein.
Yeah. So absolutely. There's many ways in which to test and see what does okay. So my answer originally was not for transient experiments. It was for equilibrium experiments. Those are the ones I wouldn't trust. For transient ones, yes, exactly. There's strategies such as this one in which you can really learn a lot about what is due to the protein and what is due to the solvent. There's different ways in which we approach this. Yes, one of the things you worry about is that about 100 nanoseconds or so, that's about the time scale in which you get sort of acoustic waves radiating from the heat spot, and you can see distortions of the signal. So that's something that you can see just by doing some control experiments on tiny, unstructured peptides. You can, sort of see what that response looks like, and that does occur. It's all much faster, than actually even our RT-jump was. At doing things lower than the folding of the protein, yes, in principle, that tells you. One of the things that our insulin project does show you is that the range over which the temperature-dependent equilibrium changes is very broad. So you may see small spectral changes that are dominated by a thermal effect, but you may just as well be looking at conformational changes within, let's say, the folded state, to adapt to the new temperature. And so it's complicated. So we've tried that experiment, for instance, to look at the switching between this native and twisted dimer form, and not having huge amount of success so far, a little bit.
All right, we have another question here from Adam Antozorski. I apologize if I mispronounced that. For the rupturing of the beta sheet, you found as an intermediate between native and twisted dimer states. Was this a local rupturing? or does it involve large-scale rotation of the beta strands away from the rest of the protein complex? How does the water actually solvate the beta sheets?
Okay. The native and the twisted form, these are both structures which have a very well folded interface. There is no water, in this interface. So the registry of the two beta sheets has slipped in rotating the angles of these helices. The whole packing of all of these aromatic side chains has switched around. It's still well packed. It's just a different packing configuration of those 5 hydrophobic side chains there at the interface. The solvation process, that's really predicted from that Markov state model to occur in the exchange between native and twisted forms. The dominant flux that in that model between the two states really involved the essentially the hydrophobic contacts largely being in place, the strands of the beta sheet losing their hydrogen bonds in water, essentially solvating them to varying degrees. So we just, it's no direct proof of that, but that's, how we interpret, the results of those simulations. Hopefully that answers your question.
Thank you. There's one other question from a member of the audience, Andreas Hugert. And the question is, quite a few peptide drug molecules associate, aggregate, and form not only dimers, but rapidly multimers and subsequently amyloid structures. What do you think of the applicability of the techniques described here to amyloid systems?
Thanks. It's an interesting question. There's a whole number of issues to answer there. Let me see. Maybe the first way to start with this is just to tell you, as far as formation of amyloid, I'd refer you to some experiments, done over several years by Marty Zanni's group, who has really looked at this process in quite some detail. In not an insulin per se, but really looked at the nucleation, the formation of oligomers, and the growth of fibrils in amyloid systems using 2D IR techniques and selective isotope labeling. And that really he's gained a lot of insight by using that strategy. Now, those are, long time scale processes. I wouldn't necessarily call them dynamics, but he really can sort of see stepwise changes, in structure. There's these other aspects to this. So I will say insulin, you probably know, has a huge propensity to aggregate. That was actually the bane of our existence, finding configurations in which we can stabilize the dimer, solubilize, the dimer, and not have a hexamer form, higher oligomers, or aggregation. It took a lot of work figuring out how buffer conditions, how pH, salt content, co-solvents, all that influence these abilities. The one sad truth is that we have an amazing ability to see when things aggregate, because it has an incredibly bright response in all of the IR spectroscopy. It immediately not just scatters, your signal, but has a very sharp feature. So there's no mystery when you start creating higher oligomers. As far as drugs go, this is an interesting question because, of course, one of the places in which this problem is discussed quite a bit for insulin is actually in the shelf life and stability of insulin drugs. So the idea is that you want to, the instability of the hexamer, and it is something you want to avoid, because that, even though that would lead to fast dissociation, they also hastens the lifetime of the degradation of the sample. I think you want to have a sample you can store for at least several weeks or a few months, if you can. That doesn't happen without real formulations to consider stabilizing oligomeric forms, but in such a way that you still keep the dimer relatively weakly bound, so it can dissociate once it hits the bloodstream. So there's a yes, even just for insulin, there's a huge business in trying to understand sort of oligomeric behavior and what influences the stability.
I don't think we have any other questions from the audience, but I have one kind of quick one, if you don't mind. In your slide where you had shown the different temperature curves for different labels, there was, I didn't quite get it. I think it started with like A19, there was a dual label, and it looked like it went up first and then down. It was kind of significantly different for the other one. I was wondering if you care to comment on what that sort of signature can tell you.
So, yes. So, well, first of all, when you see something like that, your first response should be something's wrong, because that doesn't happen. And of course, you depending on how you construct these curves, you can easily get that kind of behavior if you're taking frequency slices out of a complex multi-component system that's going up and down and positive and negative and all that. So yeah, don't take it too seriously. However, there are a handful of labels that actually do that. Under our high ionic strength conditions, there are a couple of labels that really seem to be reporting on this native twisted dimer configuration. So I showed you the example of this label B24, B25 that was right at the beta sheet interface, but it turns out there's other labels where there are two features in the loss spectrum, right? So the parts that you pulled out of these, in the loss spectrum, you can see different features rising and falling, and so there are signs that something is going on there, and we are using these types of spectral models in order to structurally interpret what's happening there. And I didn't get the chance to show it, but there's a lot of correspondence between the structures from simulation and the modeled spectrum, what we see experimentally. But there's also a handful of examples where it's really off. And of course, that leads to to questions about what the origin of that is. But my sense is, I always think that from a modeling point of view, you can never be sure that you're quite sampling the right structural configurations for everything. But yes, those are the kinds of observables, particularly if you can kinetically resolve them, that really should give you some clean insight into how different local conformational changes occur, what types of different conformational substates exist.
Thank you. And actually, we got one more question from William Jeffries, and I think this will be a good place to end. Could salts and co-solvents be used to make dissociation of dimers and possibly larger hexamer complexes occur in a generalizable mechanism across different systems?
It's a good question. Of course, well, this is a strategy many people have had going back decades. We have done some work on that problem. Well, we've done a lot of work on that problem. We have one published paper that addresses that, at least primarily in the case of DMSO as a co-solvent. We haven't, ionic strength stuff hasn't been published yet. pH hasn't been published yet. All of these matter. So the motivation, we used ethanol, and that was the original motivation, was to destabilize the dimer just enough, that it would rapidly dissociate with the temperature jump, and it did work in that respect. Of course, the behavior you see with varying amounts of ethanol changes, and not necessarily in an easily predictable manner. And in fact, different co-solvents give you different behaviors. In the case of ethanol, there's a very nice simulation study by Biman Bagchi, who has done a lot of work on this problem and comparing how water and ethanol water mixtures behave. And he's really concluded that there's considerable differences between how these two types of dimer systems dissociate, with ethanol systems having a real propensity to get ethanol into the dimer interface more rapidly and to really loosen up a lot of the aromatic packing. And so the dissociation pathways are qualitatively different than you see in pure water. We've looked at acetonitrile and DMSO extensively. What I can tell you is each solvent is different. They destabilize things in different manners. You end up with different loss of secondary structures. Everything subtle but clearly different. So every little change to the sample conditions leads to noticeable results. So that's took us a long time to appreciate how sensitive this particular sample is.
Okay. All right. So I think that we had that. Thanks for the discussion. We will thank Professor Tokmakoff one more time for a great talk and for being so gracious with his time. Now we usually end these sessions with a little bit of a student-only Q&A. So we'll ask everyone in the audience who is a faculty member or at least no longer in training to please sign off. Thank you again for joining us today. We'll also stop the streaming to YouTube, and then we can have a short, since we went over on the main Q&A. Then we can keep